This is where it all began. Billions of years before oxygen. A single breath that changed everything.
It was poison to almost everything.
Cyanobacteria didn't know what they were unleashing.
This red is their grave.
It killed the old world
And gave birth to the new.
the Great Oxygenation Event. The poison that became life.
Imagine an ancient Earth shrouded in a thick haze of carbon dioxide, methane, and nitrogen with no trace of the oxygen that would one day define life as we know it. This was our planet more than 3 billion years ago, a world locked in an anoxic embrace where the air itself was a reducing agent, fostering the earliest sparks of existence in ways utterly alien to the aerobic biosphere of today. The oceans, vast and stratified, held layers of water separated by density and chemistry, with surface waters warmed by a young sun and deeper zones rich in dissolved ferrous iron, giving them a reddish tint from suspended minerals. No blue skies reflected on these seas. Instead, the atmosphere absorbed and scattered light in hues of orange and brown, casting a perpetual twilight over the primordial soup. Life had emerged perhaps as early as 3.8 billion years ago, in the form of simple prokaryotes, bacteria and archaea that thrived without the need for oxygen. These microscopic pioneers relied on anaerobic processes to extract energy from their environment. Fermentation broke down organic molecules in the absence of an electron acceptor, producing ethanol or lactic acid as byproducts. Methanogens, a group of archaea, converted carbon dioxide and hydrogen into methane, releasing it into the atmosphere and contributing to a greenhouse effect that kept the planet habitable despite a fainter sun. Sulfate-reducing bacteria utilized sulfate ions to oxidize organic matter, producing hydrogen sulfide that permeated the waters with a rotten egg stench, though no noses existed to detect it. In shallow coastal lagoons and hydrothermal vents on the ocean floor, anoxygenic phototrophs harness sunlight without producing oxygen. Purple sulfur bacteria, for instance, used hydrogen sulfide as an electron donor, creating elemental sulfur as waste and painting the sediments in vibrant purples and greens. Green sulfur bacteria dwelled in deeper, dimly lit zones, capturing faint light to drive their metabolism. These organisms formed dense microbial mats, layered communities that trapped sediments, and built stromatolites, domed structures that fossilized into some of the oldest evidence of life, like those found today in the Pilbara Craton of Western Australia or the Gunflint Chert in Canada. The energy yields of these anaerobic pathways were modest compared to what would come later. Fermentation provided only 2 molecules of adenosine triphosphate per glucose molecule, a far cry from the efficiency of aerobic respiration. Yet, this was sufficient for a biosphere dominated by single-celled entities, where competition was fierce but limited to chemical niches. Archaea and bacteria exchanged genes horizontally, blurring lineages and accelerating adaptation in this oxygen-free realm. The planet's surface was barren of macroscopic life. No forests, no animals, only microbial films coating rocks and waters. Volcanic activity spewed gases that maintained the reducing atmosphere, while comet impacts delivered organic compounds, fueling the cycle. Methane levels were high, perhaps thousands of times greater than today, trapping heat and preventing a frozen world. The moon, closer then, drove massive tides that mixed nutrients and stirred the seas. In this equilibrium, life persisted for hundreds of millions of years, evolving slowly in isolation from the oxidative forces that would soon disrupt it. But change was brewing in the microbial world. Around 2.7 to 3 billion years ago, a revolutionary innovation appeared: oxygenic photosynthesis. Cyanobacteria, descendants of earlier phototrophs, cracked the code of using water as an electron donor in photosynthesis. This process split water molecules, releasing oxygen as a byproduct, a toxic waste product in an anoxic environment. At first, this oxygen was quickly scavenged by dissolved ferrous iron in the oceans, oxidizing it to ferric iron and precipitating out as vast deposits known as banded iron formations. These rust-red layers, alternating with silica-rich bands, blanketed the seafloor across continents like North America, Australia, and South Africa, locking away the oxygen and delaying its atmospheric buildup. For perhaps 300 million years, this balance held. Cyanobacteria proliferated in shallow waters, forming extensive mats that bubbled with nascent oxygen, but the gas remained confined, reacting locally and sparing the global biosphere. The ancient seas teemed with these innovators, their chlorophyll absorbing red light and reflecting blue-green hues that would one day color our planet from space. Yet, this innocuous innovation was a ticking bomb. As cyanobacterial populations exploded, driven by abundant sunlight and nutrients, oxygen production began to outpace the iron sinks. The stage was set for a planetary transformation that would poison the old world and forge a new one. In those final anaerobic epochs, the Earth was a crucible of simplicity, where life clung to chemical gradients and avoided the radical reactivity of free oxygen. Archaea burrowed into sediments, bacteria cycled sulfur and nitrogen, and the first hints of symbiosis emerged among them. No complex cells yet existed. Eukaryotes were still a distant dream. The atmosphere, thick with methane and ammonia, shielded the surface from harsh ultraviolet radiation, allowing fragile organic molecules to persist. Hydrothermal vents, black smokers spewing mineral-rich fluids, hosted thriving communities of chemotrophs that drew energy from Earth's internal heat independent of the sun. This era, known as the Archean Eon, spanned from about 4 billion to 2.5 billion years ago, a time when continents were just forming. Supercontinents like Valbara and Ur began to assemble, and the planet's magnetic field protected it from solar winds. Life was resilient, but fragile, adapted to a world where oxygen was absent or fleeting. Stromatolites dotted shorelines, their layered growth recording seasonal cycles in a fossil record that survives to this day. In places like the Dresser Formation in Australia, evidence of these early mats reveals a biosphere teeming yet invisible to the naked eye. As oxygen whispers grew louder, subtle shifts occurred. Some anaerobes retreated to deeper oxygen-free zones, while others began evolving rudimentary defenses. But the tipping point loomed. The Great Oxygenation Event was not a sudden cataclysm, but a gradual escalation, building over millions of years until the sinks saturated and oxygen spilled into the air. This would mark the end of the anaerobic dominion and the birth of a new oxygenated order, one that would demand adaptation or extinction. The ancient Earth pulsed with quiet potential, its microbial inhabitants unaware of the revolution they had unleashed. In the shallows, cyanobacterial blooms expanded, their photosynthetic machinery churning out the gas that would redefine life. Iron oceans absorbed the excess, forming mile-thick deposits that today supply much of our steel. But the balance was fragile. As production ramped up, the first whiffs of oxygen escaped into the atmosphere around 2.5 billion years ago, oxidizing surface rocks and hinting at the storm to come. Life had existed for billions of years in harmony with its anoxic cradle, and it seemed eternal. Until the error appeared, the byproduct that would poison the old and empower the new. The catastrophe arrived not with thunder or fire from the sky, but with a silent, insidious poison produced by life itself. Around 2.4 billion years ago, the delicate equilibrium that had sustained the anaerobic world for so long began to fracture. Cyanobacteria, those unassuming innovators of oxygenic photosynthesis had multiplied across shallow seas and coastal lagoons for hundreds of millions of years. Their mats spread like living carpets, capturing sunlight and splitting water molecules to release oxygen as a metabolic byproduct. At first, this oxygen was consumed almost instantly by the vast reservoirs of dissolved ferrous iron in the oceans. Every molecule of oxygen reacted with iron ions, oxidizing them from ferrous to ferric form and precipitating them out as iron oxides that sank to the seafloor in rhythmic layers, forming the banded iron formations that now stretch across ancient cratons in places like Western Australia, South Africa, and the Canadian Shield. These banded iron formations, alternating bands of iron-rich chert and hematite, grew to thicknesses of hundreds and even thousands of meters in some regions. They represent one of the most dramatic geological records of the Great Oxygenation Event, a planetary-scale sink that delayed atmospheric oxygen buildup for perhaps 300 million years. As long as iron remained abundant in solution, oxygen stayed trapped, reacting locally and sparing the global biosphere from immediate harm. The oceans remained stratified, with anoxic deeper waters preserving the old anaerobic communities, while surface layers hosted the oxygen-producing pioneers. But the sinks were finite. Iron concentrations gradually declined as precipitation removed it from circulation. Volcanic outgassing could no longer replenish the reductants fast enough to match the rising oxygen flux from proliferating cyanobacteria. Around 2.4 to 2.3 billion years ago, free oxygen began to escape into the atmosphere in meaningful quantities. Levels rose from virtually undetectable traces, less than 1 part per million, to perhaps 1 to 10% of modern values, though estimates vary widely depending on proxies like sulfur isotopes and chromium speciation in ancient rocks. This was no sudden explosion. It unfolded over tens of millions of years, with episodic pulses known as oxygen whiffs detected in earlier sediments as far back as 3 billion years ago. Yet the sustained rise around 2.4 billion years ago marked the true tipping point. For the dominant anaerobic organisms, this oxygen was a lethal invader. Reactive oxygen species, superoxide radicals, hydrogen peroxide, hydroxyl radicals, formed in the presence of free oxygen and attacked cellular components with ruthless efficiency. Proteins denatured, lipids peroxidized, DNA strands broke. Most prokaryotes lacked the enzymatic machinery to neutralize these toxins. Superoxide dismutase catalase and peroxidase enzymes, which today protect aerobic life, were either absent or present in rudimentary forms insufficient for survival against sustained exposure. Methanogens, sulfate reducers, and many fermenters perished en masse as oxygen infiltrated their habitats. Vast microbial ecosystems collapsed, their diversity plummeting in what paleontologists now recognize as Earth's first major extinction event, though quantifying species loss remains challenging due to the microscopic scale and poor preservation of prokaryotic fossils. Geochemical signatures bear witness to this die-off. The sulfur isotope record shows a dramatic shift. Before the event, mass-independent fractionation of sulfur isotopes, produced by ultraviolet photolysis in an oxygen-poor atmosphere, created distinct patterns in sedimentary rocks. After 2.4 billion years ago, this fractionation largely disappeared, indicating that oxygen had thickened the ozone layer enough to block ultraviolet radiation from reaching the surface. Chromium isotopes in ancient soils reveal oxidative weathering of continental rocks, a process impossible in an anoxic world. Molybdenum and other redox-sensitive elements became more mobile in oxygenated waters, further altering ocean chemistry. The climate responded violently. Methane, a potent greenhouse gas produced abundantly by methanogenic archaea, began to oxidize in the atmosphere as oxygen levels rose. Methane concentrations plummeted, weakening the greenhouse effect that had compensated for the faint young sun. Global temperatures dropped sharply, triggering the Huronian glaciations, 3 major ice ages between approximately 2.4 and 2.1 billion years ago. Some episodes may have approached snowball Earth conditions, with ice sheets extending to equatorial latitudes and sea ice covering much of the oceans. Under this frozen shell, surviving anaerobes retreated to deep subsurface environments, hydrothermal vents, or anoxic pockets, beneath the ice, clinging to chemical energy sources untouched by oxygen. The oceans themselves transformed. As ferrous iron precipitated out, deeper waters became less reducing, allowing oxygen to penetrate farther. Sulfide-rich zones contracted, and new redox gradients formed. In surface waters, oxygen-tolerant microbes began to proliferate, while deeper layers preserved pockets of the old anaerobic world. The banded iron formations themselves began to decline after about 2.3 billion years ago as the iron sink saturated and oxygen could no longer be fully scavenged. This was the oxygen catastrophe in full force, a planetary poisoning event driven by life's own success. The gas that would one day power complex respiration acted first as a sterilizing agent, wiping out lineages that had ruled unchallenged for billions of years. The extinction was not dramatic in the fossil record, no dramatic iridium layers or shocked quartz, but its geochemical fingerprints are unmistakable. Sulfur isotopes, iron formations, chromium mobility, and glacial deposits all converge on this singular truth. Oxygen killed the old world to make room for the new. Yet, even in the midst of collapse, seeds of survival were sown. Some anaerobes developed primitive defenses against reactive oxygen species, enzymes that would later become universal in aerobic life. Others migrated to refugia, deep sediments, hydrothermal systems, or isolated anoxic basins, where they persist to this day as extremophiles. The stage was cleared, the old players removed, and the path opened for those few that could turn poison into power. The Great Oxygenation Event had begun its work, not as an end, but as the most profound beginning in Earth's biological history. The catastrophe had done its work. Vast swaths of the anaerobic biosphere lay in ruins, their lineages reduced to scattered survivors, clinging to the deepest, darkest refugia. Yet from this devastation emerged the first faint glimmers of a radically different future. The oxygen that had poisoned so many now lingered in the surface waters and atmosphere, creating new ecological opportunities for those few organisms capable of harnessing it. The Great Oxygenation Event, though destructive, had opened a door that would never close again. The survivors were a resilient minority. Some archaea and bacteria retreated to anoxic sediments, hydrothermal vents, or stratified ocean basins where oxygen could not penetrate. These holdouts preserved ancient metabolisms, methanogenesis, sulfate reduction, iron reduction, that persist in extreme environments to this day. Others, however, began to adapt. Primitive versions of superoxide dismutus and catalase enzymes appeared in certain lineages, neutralizing reactive oxygen species and allowing limited exposure to the new gas. These molecular defenses, initially crude, marked the first steps toward aerobic tolerance. The true revolution came with aerobic respiration. Oxygen, once a toxin, became the ultimate electron acceptor in a metabolic pathway far more efficient than any anaerobic process. Where fermentation yielded only 2 molecules of adenosine triphosphate per glucose, aerobic respiration produced up to 38, a staggering 18-fold increase in energy harvest. This energetic windfall allowed cells to grow larger, reproduce faster, and invest in more complex structures. Organisms that mastered aerobic metabolism gained a decisive advantage in the newly oxygenated surface waters. Perhaps the most transformative adaptation was endosymbiosis. Around 2.1 to 1.8 billion years ago, an archaeal host cell engulfed an aerobic alpha proteobacterium. Instead of digesting it, the host retained the E-al bacterium as an endosymbiont. Over time, this relationship became permanent. The bacterium evolved into the mitochondrion, transferring most of its genes to the host nucleus while retaining a small genome for core respiratory functions. The host cell gained access to mitochondrial aerobic respiration, unlocking unprecedented energy production. This event, occurring once in evolutionary history, gave rise to all eukaryotes, organisms with complex cells containing nuclei and organelles. The first eukaryotes were likely simple protists, single-celled forms that combined the archaeal host's complexity with the mitochondrial power plant. These cells could now support larger sizes, more intricate cytoskeletons, and dynamic behaviors like phagocytosis, the engulfment of other cells for food. Predation emerged as a viable strategy. Early eukaryotes began consuming bacteria and other protists, accelerating nutrient cycling and driving further innovation. Some lineages developed chloroplasts through a second endosymbiosis, engulfing cyanobacteria and transforming them into organelles capable of photosynthesis. These photosynthetic eukaryotes, ancestors of algae and plants, further boosted oxygen levels while creating new food webs. The oceans, once dominated by microbial mats, began to host more diverse communities. In oxygenated surface layers, eukaryotic protists formed loose aggregations, some developing colonial forms that foreshadowed multicellularity. Filamentous algae and early red algae appeared, their pigments absorbing light in deeper waters. Predatory protists with flagella or pseudopods hunted smaller cells, introducing selection pressures for armor, speed, and evasion. The redox gradient in the oceans sharpened. Surface waters became mildly oxygenated, while deeper zones remained anoxic or euxinic, sulfide-rich, preserving pockets of the old anaerobic world. Geochemical evidence from this period shows the transition clearly. Banded iron formations largely ceased after about 2.3 billion years ago, as surface iron was depleted and oxygen persisted. Molybdenum concentrations in black shales increased, indicating more oxygenated waters that mobilized this element. Sulfur isotopes stabilized, reflecting a thicker ozone layer that shielded the surface from ultraviolet radiation and allowed more stable organic preservation. Glacial deposits from the Huronian interval tapered off by 2.1 billion years ago, suggesting greenhouse recovery as oxygen levels stabilized and new carbon cycles emerged. This era, bridging the Archean and Proterozoic eons, saw the biosphere cross an irreversible threshold. Energy abundance from aerobic respiration enabled experimentation with size, shape, and complexity. Cells could now afford elaborate membranes, internal compartments, and cytoskeletal networks. Sexual reproduction likely evolved around this time, shuffling genes more efficiently than horizontal transfer alone. The stage was set for the slow climb toward macroscopic life. In the quiet depths of those ancient seas, the first true titans began to stir. Not yet the colossal predators of later eras, but larger, more active single-celled organisms that exploited the new energy landscape. Eukaryotic grazers cropped microbial mats, while early predators pursued them in three-dimensional water columns. The oxygen revolution had cleared the field of the old giants, microbial empires, and replaced them with the seeds of new ones. From the ashes of extinction rose the potential for complexity, mobility, and eventually the diverse marine ecosystems that would dominate the planet for billions of years. The Great Oxygenation Event had not merely changed the atmosphere, it had rewritten the rules of life itself. Energy flowed more freely, cells grew more ambitious, and the path to multicellularity opened wide. The titans of the ancient seas, armored, swimming, hunting forms, were still distant, but their foundations had been laid in the quiet wake of catastrophe. Oxygen, the poison-turned power source, had birthed a world capable of wonders that the anaerobic Earth could never have imagined. The oxygen revolution had cleared the field, leaving behind a biosphere forever altered. With aerobic respiration now possible, energy flowed in abundance, unlocking potentials that the anaerobic world could never have supported. Cells that once scraped by on meager fermentation yields could now afford elaborate structures, larger sizes, and dynamic lifestyles. The path from microscopic simplicity to macroscopic complexity had opened, and the ancient seas began to host the first true titans, organisms capable of active movement, predation, and ecological dominance. In the Proterozoic oceans, roughly 2.1 to 1 billion years ago, eukaryotic protists diversified rapidly. These early complex cells, equipped with mitochondria, cytoskeletons, and endomembrane systems, experimented with new forms. Some developed flagella or cilia for propulsion, allowing them to swim through oxygenated waters rather than drift passively. Others evolved pseudopods or amoeboid motion, engulfing prey through phagocytosis. Grazers cropped microbial mats, while predators pursued smaller eukaryotes, introducing arms races that favored armor, speed, and evasion tactics. The first hints of multicellularity appeared. Loose colonies of cells that divided labor, with some specializing in photosynthesis or reproduction. Filamentous forms like red algae precursors spread across shallow seafloors, their pigments adapting to capture light in deeper oxygenated layers. By approximately 1.8 billion years ago, eukaryotic diversity had exploded. Fossils from sites like the Gripania spiralis in Michigan and the Nama group in Namibia show macroscopic algae reaching centimeters in length, simple ribbons and spirals that represent some of the earliest evidence of large, multicellular eukaryotes. These organisms exploited the new energy surplus to grow beyond the diffusion limits that constrained prokaryotes. Larger size demanded more efficient nutrient uptake, leading to innovations, like vacuoles for storage and transport systems for internal circulation. Sexual reproduction likely emerged during this period, allowing genetic recombination that accelerated adaptation and reduced the accumulation of harmful mutations. Predation became a dominant force. Early eukaryotic hunters developed structures for capturing prey, grasping appendages, stinging organelles, or simply overwhelming speed. In oxygenated surface waters, food webs grew more complex. Primary producers, photosynthetic protists and algae, formed the base, grazed by heterotrophic protists, which in turn were hunted by larger predators. This trophic structure foreshadowed the ecosystems of later eras. Redox gradients in the oceans sharpened further. Surface layers supported aerobic communities, while deeper, still anoxic zones preserved pockets of anaerobic life. Sulfidic conditions in some basins created black shales rich in organic carbon, preserving delicate microfossils that reveal this transition. Around 800 to 600 million years ago, the stage was set for even greater leaps. Oxygen levels continued to rise, perhaps reaching several percent of modern values, driven by proliferating photosynthetic eukaryotes and declining burial of organic matter. The cryogenian glaciations, two major snowball Earth episodes around 720 and 635 million years ago, tested this emerging biosphere. Global ice cover may have reached equatorial latitudes, but open water pockets and subglacial lakes provided refugia. Post-glacial warming triggered rapid diversification. The Ediacaran biota appeared around 635 million years ago. These soft-bodied organisms, preserved in sites like Mistaken Point in Newfoundland and the Ediacara Hills in Australia, represent the first widespread macroscopic multicellular life. Dickinsonia, Spraguina, Charnia, and others grew to tens of centimeters, some forming quilted mats, others frond-like or disc-shaped. Debate persists over their affinities. Some may be early animals, others fungal or algal colonies, or entirely extinct lineages. But their presence marks a threshold. Oxygen had enabled the energy demands of larger bodies, diffusion-based respiration, and simple movement. Some Ediacarans show evidence of mobility, grazing trails, and possible predation, hinting at emerging ecological complexity. The Cambrian explosion, beginning around 541 million years ago, built directly on this foundation. Oxygen levels stabilized or rose further, supporting the burst of animal phyla. Trilobites, brachiopods, anomalocaridids, and early chordates appeared with hard parts, complex eyes, and active lifestyles. The first true apex predators, creatures like Anomalocaris with grasping appendages and compound eyes, dominated shallow seas, hunting in 3 dimensions. Armor evolved as defense, burrowing as escape, swimming as pursuit. Food webs became intricate, with herbivores, carnivores, and scavengers filling niches that the anaerobic world could never have sustained. This progression, from eukaryotic protists to Ediacaran experimenters to Cambrian titans, was fueled by the energy surplus of aerobic metabolism. Larger sizes demanded more oxygen, but also allowed more efficient gas exchange through gills or diffusion across thin tissues. Active lifestyles required higher metabolic rates, which oxygen enabled. Predation drove morphological innovation, segmentation, appendages, sensory organs. The ancient seas, once quiet microbial realms, now teemed with movement, competition, and diversity. The titans of these waters were not yet the colossal marine reptiles or giant fish of later eras, but their foundations had been laid. Early predators chased prey through oxygenated currents, grazers cropped algal meadows, and scavengers recycled nutrients. The Great Oxygenation Event had not merely changed the chemistry of the planet, it had rewritten the possibilities of life. From the ruins of anaerobic simplicity rose the potential for complexity, mobility, and ecological dynamism. The path to the diverse marine ecosystems that would dominate Earth for billions of years had been cleared, and the first true giants of the deep-time oceans had begun to emerge. The Great Oxygenation Event left an indelible mark on Earth's history, one whose consequences echo through every breath we take today. What began as a planetary poisoning event triggered by life's own byproduct ultimately became the single most transformative moment in the biosphere's 4 billion-year story. The oxygen that had extinguished countless anaerobic lineages did not merely survive the catastrophe, it reshaped the rules of existence itself, setting the stage for complexity, diversity, and the rise of macroscopic life that would culminate in the ancient seas and beyond. The legacy of this event is woven into the very fabric of eukaryotic biology. Every mitochondrion in every eukaryotic cell traces its ancestry back to that single endosymbiotic event between an archaeal host and an aerobic alpha proteobacterium. This partnership, forged in the aftermath of the oxygen rise, provided the energy surplus that made complex cells possible. Without it, the diffusion limits of anaerobic metabolism would have kept life forever microscopic, confined to thin films in simple colonies. The mitochondrion's efficiency, yielding up to 38 molecules of adenosine triphosphate per glucose compared to fermentation's meager 2, unlocked the potential for larger genomes, intricate cytoskeletons, internal compartments, and dynamic behaviors like phagocytosis and motility. This energetic foundation enabled the transition from prokaryotic simplicity to eukaryotic sophistication, a leap that occurred only once in evolutionary history and gave rise to all plants, animals, fungi, and protists. The oceans of the Proterozoic and into the Phanerozoic bear direct witness to this inheritance. The Ediacaran biota, emerging around 635 million years ago, represented the first widespread experiments in macroscopic multicellularity. Soft-bodied forms like Dickinsonia, with their quilted surfaces, grazed microbial mats or absorbed nutrients directly. From the water column, Charnia, with its frond-like structure, may have hosted symbiotic algae, capturing light in low oxygen conditions. These organisms, some reaching tens of centimeters or even a meter in length, depended on the elevated oxygen levels that allowed diffusion-based respiration across greater body sizes. Their appearance after the cryogenian glaciations suggests that post-glacial nutrient pulses and stabilizing oxygen further accelerated the shift toward complexity. The Cambrian explosion, beginning around 541 million years ago, built explosively on this foundation. Oxygen had reached levels sufficient to support active lifestyles, hard parts, and predation. Trilobites developed compound eyes and segmented bodies for efficient movement. Anomalocaris and other radiodonts became the first true apex predators, with grasping appendages, circular mouths, and swimming capabilities that allowed them to hunt in three-dimensional space. Brachiopods, sponges, and early mollusks filled filter-feeding and grazing niches. The energy demands of these innovations, mineralized shells for protection, nervous systems for coordination, muscles for propulsion, were met by aerobic respiration. Without the oxygen revolution, such metabolic extravagance would have been impossible. Even the giants of later eras owe their existence to this ancient shift. The marine reptiles of the Mesozoic, Ichthyosaurs, plesiosaurs, mosasaurs evolved from terrestrial tetrapods that themselves descended from Devonian fish, whose ancestors traced back through Cambrian chordates to the first oxygen-breathing eukaryotes. Modern whales, sharks, giant squid, and the colossal filter feeders of today's oceans all inherit the metabolic toolkit refined in the wake of the Great Oxygenation Event. The aerobic pathway, once a desperate adaptation to a toxic gas, became the universal engine of animal life. Geochemical records reinforce this narrative. The disappearance of mass-independent sulfur isotope fractionation after 2.4 billion years ago signals the formation of a protective ozone layer, shielding surface life from ultraviolet radiation and allowing more stable organic preservation. Chromium and uranium isotopes show oxidative weathering on continents, mobilizing elements that fed into evolving nutrient cycles. The long Proterozoic boring billion, from roughly 1.8 to 800 million years ago, saw relatively stable oxygen levels, giving time for eukaryotic diversification to accumulate innovations that would later explode in the Ediacaran and Cambrian. The paradox remains profound. The same oxygen that caused the first mass extinction also enabled the biosphere to transcend its microbial origins. It poisoned the old world to birth the new, clearing space for energy-hungry innovators that could exploit the gas rather than flee it. Without this crisis, there would be no mitochondria, no eukaryotes, no multicellularity, no predation, no Cambrian explosion, and no lineage leading to the complex marine ecosystems that dominated Earth for billions of years. The titans of the ancient seas, the armored hunters, the swift swimmers, the colossal filter feeders, were not inevitable. They were made possible by a catastrophe that nearly ended life, only to remake it on a grander scale. Today, every breath we draw carries forward the legacy of that distant event. The oxygen in our lungs, the energy in our cells, the diversity of life around us, all trace back to the moment when cyanobacteria released a waste product that reshaped a planet. The Great Oxygenation Event was not merely a chapter in Earth's history. It was the pivot on which the entire story of complex life turned. From the ruins of an anaerobic dominion rose a world capable of wonders, where the poison of yesterday became the breath of tomorrow. The ancient seas, once silent and simple, now teem with the descendants of those first oxygen-breathing pioneers. And in their silent depths, the echoes of that primordial catastrophe still resonate, reminding us that even the greatest extinctions can be the seeds of the greatest beginnings.